questions. I'll keep an eye on the chat log if I can. And yeah, so my name is Tracey and I am a PhD student in Stellenbosch. I have done a lot of immunofluorescence staining. Most of my master's was made up of fluorescence imaging, and this is both on tissue and cells. So more recently on cells, but a lot of tissue work. So I just wanted to briefly touch on the principle of immunofluorescence. So I hope that you guys can see, but I know that the previous talk kind of mentioned it, but I want to just also mention the pros and cons of using both indirect and direct staining. So with direct staining, you'll be using a conjugated antibody. So your fluorophore is already attached to your antibody. So the pros of this is a quicker stain because you won't have to waste the time in using a secondary antibody. However, with indirect staining, it's a two-step process where you'll first stain with your primary antibody, and then following that, you'll stain with the secondary antibody, which has got the fluorophores attached. The pros of using indirect staining is that you can choose the different wavelengths that you want to use. So you're not restricted with a specific antibody having a specific wavelength. So you can then use that antibody for multiple different types of staining. I find it most beneficial to use the indirect, even though the direct does seem a bit quicker. But if you are going to use a specific stain for one thing the whole time, I'll give an example later on with the images, then you can use that direct form. And then, so those are the main two types of things to consider. And then also, the setting up of your stains does take a little bit of time, and you have to be patient with it, because if you don't set up your stains adequately, when you're actually trying to run all of your samples, it's not going to look as good and you're not going to get the results that you want. So in that sense, if you are using multiple stains and multiple antibodies, I would suggest using single stains initially and making sure that all of them work with the secondary that you are using, and that there isn't any autofluorescence that the secondary is staining before adding them together. So you'll first do your single stains with your multiple different antibodies, and then you'll test them out together and make sure that you are seeing the thing that you are supposed to be seeing. Another important thing is to make sure that you actually know what you are looking for. There are many times where I have helped people where they don't know where the protein of interest is that they are looking for, and that can be very problematic because you can't know where everything is yourself, and you need to know what you are looking for. So also when you're setting up your stains, you need to have positive and negative controls because of the autofluorescence that does occur. You need to do stains where you will stain only with your primary antibody, and you'll stain it alongside something like Hushed or DAPI, so that you can see that your primary antibody doesn't give off fluorescence. And you'll do the same thing with your secondary antibody, because the secondary antibody won't have a primary to bind to, therefore you shouldn't be seeing any fluorescence. And that you can get a good indication whether your antibodies are good, and you also can set up your different methods of imaging to make sure that if there is autofluorescence, that you take that into account. You also need a PBS control because some tissue has autofluorescence. So there you'll only have PBS, you won't have a primary or a secondary stain or antibody, and you'll be able to see your autofluorescence. It's useful to have the Hushed or the DAPI because of focusing of the image. That's the main reason that we still put that on with all of our stains. There also is a difference with paraffin embedded and frozen samples. So your paraffin embedded samples will have to go through the dehydration process, whereas your frozen samples don't. So just keep that in mind. And then it's also very important to consider permeabilization. I always use permeabilization. It's just a step that I always account for. Whether you're doing internal protein or external, it's always just useful. And then another thing that also helps with the problem solving is adequate washing. I know it's like a long step and it's boring, but I always do 5-minute washes that are 3 of each, and also I add tween to my PBS washes so that there's a little bit of detergent in there, and that allows for the whole process to clean up quite nicely. And then let me just check here quickly, make sure. Okay, I'm going to answer that question just now. Then I just want to, some of the difficulties that we face with the immunofluorescence is when you're trying to do multiple different antibodies at the same time, one of the things is that you can find that there's not enough secondaries. So you have to set your antibodies up in a way that you'll be able to see all the different proteins separately, so that you have a clear indication of what is what. So that is limiting in that you can't just stain for everything at the same time. However, you can use the same wavelength on different proteins if you know exactly where they are going to be and you can differentiate between the two. So for example, in this image over here, this is a muscle tissue and the green borders are stained with 488 and the internal fiber type is stained with 488. And we know definitely that the border and the fiber type are different, but because we know the distinct location of each, we can then stain with both with 488, and that allows us to keep open a different wavelength, like we're using 555 for the satellite cells that are focused over there. So that does open it up. And as the previous talk mentioned, sometimes you are restricted with the host of antibodies that you're using, and ideally any stain you'd want to use different hosts because you don't want your secondary to bind unnecessarily. But there is the option to use the same host where you would block in between. If you guys would like the protocol that I have used in the past, you're welcome to take down my email address and I'll happily email it to you. We have tried and tested it, and it's just a whole lot of patience in the staining. And even if we block and stain for 3 days, it's better than rushing through the process and not having a nice image in the end. Another thing that we struggle with is with the immunofluorescence, some antibodies are really sensitive to freezing. So with the frozen tissue samples, we'll section and then store the samples and stain them later on. And some antibodies literally do not show up if you have frozen them, the tissues, before staining them, which can be very frustrating to take the time to figure that out. So I would always try and use it as fresh as possible and try section and stain at least. You can freeze it overnight and stain it the next day. That would be optimal, but try not to leave them in the freezer for too long if you've got a very sensitive protein. We do get a lot of autofluorescence, which Lisa will discuss in her talk, and I'm sure the recordings will become available. But these things you can compensate for on the microscope and on the settings, and that's why we use the controls that we use. The 488 is one of the antibodies, the green, that does bring up a lot of autofluorescence. So it's just another thing to take into consideration. Yeah. So if you guys had any questions for me, I'm happy for you to just butt in and ask me questions that are bugging you. So I see the first question is about how things change with immunohistochemistry. The whole staining process is not much different. So I find that the easiest way to stain for cells is to actually grow them on a cover slip, because then you can remove the cover slip and stain them as you would normally. So yeah, that's the main difference, and everything is the same. You wouldn't do the whole dehydration and everything. You would treat it the same as frozen cells, frozen tissue. But yeah, everything is the same with that. Does anyone else have any questions for me? So one of the other issues that we might have is if the tissue or cells wash away. So you obviously can't see the cells while you're staining them, and then it's an awful process if you've done the whole staining situation and then you go to image and they've washed away. So that's the importance of fixing. So you'll always fix as your first step. I use paraformaldehyde to fix, and I do that for 10 minutes. And I do the same thing with the cells. But if you do have a more harsh stain, this works with both immunofluorescence and normal immunohistochemistry, is you can first coat your cells or your plates with polyallysine, which makes the tissue and the cells stick much more securely, and there's less chance of washing away. You do have to, although, consider autofluorescence with that, so you'll need to test that as part of your initial control steps. Does anyone else have any questions? You're welcome to turn your microphones on and ask me questions.
I wondered about the acoustic embedding that will help to get much thinner sections in the range of 1 micron sections. Have you used that before?
I personally have mainly worked with frozen tissue. It's just my preference, but I do know that you can get much thinner sections with paraffin embedding, and you also can store your tissue for a lot longer when you do paraffin embedding. I know that it's a longer process once you have done, when you to start your staining, because you'll have to dehydrate the tissue and clear off all the wax and the paraffin. But I haven't heard of any extra issues with using paraffin embedded, but you can definitely get the tissues a lot thinner. Anyone else have a question?
Okay, I have a question. So for my study, I want to collect samples. We're going to collect tissue samples from animals.
Okay.
But yeah, I've never done this one before. We want to use the fluorescence in situ hybridization method to basically visualize IP10 and interferon gamma.
Okay.
Is it better to actually then, because we're going to basically get the samples from an abattoir, so it's fresh, but will it actually be better then to freeze it and then work from that, or would it be better actually then to do paraffin embedding before we actually do the fluorescence in situ hybridization?
Okay. I personally don't have any experience in situ hybridization. There's a lab that does do that entirely at UCT. I can look up and see if I can get the contact for you, and they can potentially give you a better answer. We did have a meeting with them at the beginning of last year because we were going to do a stain like that, and they didn't have any issues with either frozen or paraffin embedded. I will write it down, and I can see if I can find the contact details for you of the people that we met with, and they can potentially help you out with that.
Thank you.
No problem. Okay, Does anybody else have any questions?
Maybe I can ask another question.
Okay.
Question in terms of selecting your antibody or checking your antibody. I found with 2 crocodile species that the antibodies didn't give me the same result. So is that quite an issue to play around in the beginning to find the specific antibody?
Yes, that's actually a very big issue that we find, especially if it's a newer protein or a newer model. We did have a study here where we stained for a certain protein with a specific antibody, and we got the results that we got, and then we went to visit another lab, and they visualized that protein in a completely different area. So that's why I say that it's very important to know where the protein of interest is that you are looking for. It just becomes more difficult if it's a new protein. But I would actually, if you've got a range of different antibodies, I would recommend testing them first, because you'll also have different reactions with the secondary, and some of them are manufactured to a specific antigen or specific part of the protein and will therefore show up differently. So if you have access to multiple antibodies, I would test them first and see which one you're getting the results that you are looking for. Basically, one of the most important things with immunofluorescence is you have to have a lot of patience in setting up the whole stain, because if you don't set it up adequately, you're not going to get the results that you're looking for, and you're going to end up wasting a lot of time staining all of your samples and then not getting the results that you want, or any results at all. So yes, I would play around with different antibodies. And also another thing that's really important when you're setting up your immunofluorescence is to do multiple dilutions. So your antibodies will come with a recommended dilution. I would go above and below that dilution, and you also would like to do that with your secondary antibody. Once you've done it, it's done, and you can use it like that forever, and with the secondary antibody, it's done. But finding that optimal concentration is going to give you the best results ultimately. Does anyone else have any other questions? Are you guys all actively doing immunofluorescence, or are you just planning?
Well, we've done one study, but obviously I didn't.
Your signal is very bad, so I can't hear you.
Anything that you did it. So yeah, I'm on a farm with very bad signal. So.
Okay, it might be easier to just post in the chat because I can't hear your actual questions. Okay, so it looks like a lot of you are planning. So I'm happy to send you guys protocols if you want. Just let me know. My email address should be as part of the invite. So if you need anything from me, if you have questions at any later stage, I'm happy to answer any questions that you might have. Even if you're doing flow cytometry or cytometry, using the antibody information is just also valuable for that. Yeah, so it sounds like most of you are in the planning phase. So what I have suggested is quite important to just consider when you are in that planning phase is the setting up of your stain is the most important part. And while it might take a few weeks, once you have it, you are not going to have any issues further from that, hopefully. Yeah, so yeah, that's basically if you have any other direct questions. Let me quickly show you. I don't know if you can't see, let me know. So this specific stain here, when I was saying the indirect and direct, the border of the fibers, we use the direct method where it's a conjugated antibody, and all of the other stains on the image are indirect. So we always wanted this borderline one to be green, and we will work around it with any of our other fluorescent stains. So that's a method that can also be used. What's nice about that antibody, which you can potentially use in the future, is that that antibody is actually grown in plant, so it's very affordable. It's called a wheat germ antibody. It goes very far, and it's a very quick stain. It takes about 20 minutes to stain the entire thing from primary to secondary, and then it gives you a lot of time for your other antibodies. Yeah, so most of the things that I'm saying is just planning, do your single stains, your positive and negative controls, and your different concentrations. Like every time you set up a stain, I would do that. So in terms of the schedule, when you're setting up a stain, every single stain takes 2 days. I would always recommend doing primary overnight. I find the best results if you do it that way. So everything takes 2 days, but you can set up all of your single stains and everything at the same time. So I would do multiple sections, and I would do concentrations, like different concentrations like that, and then do that with everything. So because the first step that you'll do is look at your different concentrations, because you don't know which one is actually going to show up. And then, so I would take those 2 days to do different concentrations of your primaries and your secondaries. And thereafter, I would do single stains to make sure that the concentration you are using doesn't have autofluorescence. So that can, if everything goes according to plan, that can take a week just for your single stains and your concentrations. And then I would potentially do a second week where you are looking at the combination of your antibodies together. And then you should be good to go after 6 solid days. If you've got no issues. But sometimes it does take a bit longer to get the optimal concentration and optimal sectioning and everything like that. But the quickest is probably 6 days, and then going from that. Okay. I don't know if you guys have any more questions for me. I'm happy to sit around for a little bit, and otherwise I'm pretty sure you can leave. But yeah, if you've got any more questions, I am here to answer them as somebody who's actually working in the lab constantly with the staining.
I'm sorry, I might have a question. So I'm working on developing a biosensor where we immobilize primary antibodies onto the electrode. And then I'm planning on using the fluorescently tagged secondary antibodies directly onto those primary antibodies to see whether the immobilization was successful. But I'm not exactly sure what I could do as a control. I was thinking of immobilizing BSA and then using the same fluorescently tagged secondary antibodies on top of that, because theoretically it shouldn't bind. But I'm not sure whether you have a different suggestion or whether you've seen, I don't know, something similar. I just don't know how to do a control because I want to, yeah.
So I would do exactly that. You can either just do a BSA, and so I would do BSA with your secondary, and then also what was I going to say? BSA and your secondary, and then also BSA with no secondary.
Okay. Okay. So
And also
like
then,
everything.
yeah, so you'll have just BSA, no primary, no secondary, and then you'll have BSA with your secondary, and you'll have I've lost track, just your primary, and then instead of using a secondary, you'll use BSA, because then you can see if anything autofluoresces.
Perfect. Okay. Thank you.
Yeah, and that's if you're going to use BSA, if you're using BSA to dilute your proteins, I mean your antibodies, then use that, or else you can also use PBS.
Yeah, I'm using PBS and goat serum.
Okay, so you don't even need to use BSA then. You can use PBS. As a recommendation, just something to consider, you don't need to do it. I used to put my antibodies in PBS, and I've started actually putting them in I think 1% BSA.
Okay.
Yeah, because the BSA acts as a blocking, so it blocks the antibodies, the antigens that are available while you are staining. So I feel like there's less autofluorescence and less non-specific staining.
Okay. Thank you. And is BSA similar to using goat serum?
Yes. So in the whole process with the staining, you'll block first. I don't know how it works with your method, but with tissues and cells and things, we block first, and that's where we use the serum.
Okay. Okay. Perfect.
Yes, so the serum will coincide with the secondary, so it blocks the non-specific stain, the binding.
Okay. Thank you.
Yes, no problem. Okay, so if anyone else has any questions, then you're welcome to stay. Else, yeah, you're welcome to leave. I hope that was helpful. Yeah.
Thanks very much.
Pleasure.
I didn't get your email. Will it be on the list?
My email should have come from the invitation to the meeting. So it is 19687052 @sun. Yeah.
Okay.
Yeah, I sent out the invitation this morning, so it should be there.